Hello once again, everyone. It's a big pleasure to be here with you, and I'm really thankful to join to get this information and knowledge all together. As you know, that oocyte vitrification, embryo vitrification, and worming now in the biggest trend of an IVF field, while social freezing, egg banking, also the egg donation increasing dramatically, and of course that all of us want to get uh, better result, better outcome in our lab, high survival and high blastocyst rate, and mostly that many embryologists and lab getting a problem or suffering with the survival or many of the cleavage arrest. So under this perspective, of course, we were trying as an we are an egg bank, we are working with many donor eggs, so we have more possibility for the research side as well. And the last 2 years, we were working hard, especially for the vitrification and warming process. We try to establish an easy process for everyone, which will increase efficiency, survival, and outcome as well. So, but in aside, when we were doing that, we want to go the deeply on the process as a metabolic, also transcriptomic way, and to find out each protocol and each process, what they are bringing us as an advantage and disadvantage. So I will start a very important diagram from the Joyce Harper and colleagues, so which they prepared. While, as you know, we started everything with the slow freezing. Even the slow freezing were important because we were keep the material as a cryopreserved, and we can save the future of people by this way. So that's why we accepted a slow freezing. It was a long process. There was a lot of difficulties, a low efficiency, but still we were using that years and years. And then vitrification process, it was a game changer and changed our life. It make more easier, more efficient. But of course, when we accept this one, as we are accepting many new technologies and techniques in the lab, we accept it easily. And still, there was a lot of question on the background of vitrification process. So mostly when we are getting some hypothesis or some new techniques. So what we should do firstly in our lab. So firstly, of course, this hypothesis or research should be analyzed very well as in a background. Then when we are accepting as a scientific way or theoretical way this hypothesis, we should make immediately as an animal model. So after that, we should test also that and human model as well. And when we have some test scale on the as a clinical IVF study, then we have to enlarge this study multi-clinical study. And finally, we have to calculate as a clinical also the financial perspective of effectiveness of this techniques or new achievements. So that is how we should apply and accept to our laboratory. So that's why there was many question also as a vitrification process, and still several studies show as the frozen embryo transfer also can give some higher risk for the cancer on the childhood stage, which we are coming directly from the frozen embryo transfer. So of course, this is an hypothesis, but still under this hypothesis, there is a big question if we are doing something wrong or everything is right for the regular vitrification process. So from this perspective, of course, there was no enough data for the metabolic and the transcriptomic way after the vitrification process. That's why we focus on this. Of course, metabolic data to get metabolic result much easier than transcriptomic, while on the transcriptomic result, we have to work for single gene basis of RNA analysis. And you know, even as a study, if we check that, you will find out several studies which is very difficult to get from the single cell to get RNA and to make the gene expression of it. So as you know, the slow freezing were focused on mostly to ice crystal formation. and then a regular vitrification, which we are keeping 12 to 15 minutes in equilibration solution, was the glass-like state. And then, of course, we just get for ultra rapid vitrification, which is giving us immediate glass-like state. So that means we are improving the process as a timing and the efficiency. So as an ultra rapid vitrification, especially for the oocyte base, it's not easy to find many studies, of course, but there are several studies which is really effective. And those studies shows as a clinical base between standard vitrification and ultra rapid vitrification, there is no significant difference on the morphological base. And compared to standard vitrification and ultra rapid vitrification as an advantage, they find out ultra rapid vitrification can reduce osmotic stress, also did not disturb the meiotic spindles, and also can increase the blastocyst rate, not because of the technique, but because of the high survival rate. So more survived oocyte, of course, give potentially more blastocyst. As a one-step oocyte and blastocyst rate rotation and warming, actually how we are doing that. Here, basically the brand of media, it's not a question, mostly that which we analyzed and which we built the protocol, the base of 1 molar sucrose. That means any media who is containing 1 molar sucrose as thawing solution that will be available to use for ultra rapid warming. So once we have frozen oocyte in a straw, just we are preparing the media with pre-warmed, minimum 1 hour pre-warmed 1 molar sucrose of TS solution, thawing solution in incubator, and of course only 37 Celsius without gas, and pre-warm Petri dish. Doesn't matter which dish, 4-well dish, central dish, depending on. But here, for ultra rapid, I will strongly recommend for the 4-well dish, because 4-well dish have a low diameter of the surface, which the timing will be very critical. As I say, the 1 minute you should keep in the TS solution. So to find and to follow oocyte in a 4-well dish will be much easier than central dish and 60 millimeter or 30 millimeter of the dish. Of course, also you can use brand Petri dishes as Cryotech, Kitazato, different brand has such Petri dishes. But here is the point that for us, 60 seconds is a very critical. While why 60 seconds? That is basically how we built this protocol, because in the 60 seconds between the cryoprotectant and water change on the vitrification process, and also for warming process, that cryoprotectant is going out, water is entering again back, and oocyte or embryo is expanding back. So that's why that 60 seconds is a very critical. Once you put the straw into the TS solution, you will just wait, oocyte will disattach from the straw, and immediately you should start the timer. And the last 2-3 seconds, you need to just take oocytes from the TS solution and transfer culture dish. So I'm recommending also to use a culture dish with high protein concentration. So mostly one-step medias including the high percentage of albumin, that means protein. But if not, you can always add additional 10% of the protein to increase. So that's helping to you to control osmotic pressure much better. So basically, when you thaw with ultra rapid warming, what you will notice different than regular warming. So you will see that oocytes are looking much closer to fresh oocytes than regular vitrification and regular warming. So this is the advantage of ultra rapid warming. So we are keeping exactly the 300 microliter of TS solution, because here we are keeping only 60 seconds, and now the exact temperature control and optimization will be very important for us. So the micro drop technique, it's a risk is still for ultra-rapid vitrification. Of course, the shorter rewarming always will be reduced recrystallization. So this will be the advantage of the technique. So I took here, there are several images. So those oocytes freeze by ultra-rapid vitrification and warm again by ultra-rapid warming. As you see the difference then, so of course there are different type of oocytes. Some of them even have a morphological abnormality, but still here as a survival, you can see that they look like more closer to fresh oocyte because basically as we have many partner clinics which they are using our frozen oocytes. So basically that for regular vitrification and warming, also we are facing on that. We saw the long time, even after warming 1-2 hours, still some kind of granulation in the cytoplasm. And when we do fertilization during ICSI, so we don't see exactly zona permeability and resistance. So the ICSI pipette is going really easy for the injection, and we cannot break exactly the cytoplasm as we want. But in ultra rapid forming, that you will have totally different experience on it. As the clinical studies, of course, there are several clinical studies which is giving information about ultra-rapid vitrification and warming. As Lieberman has study for compared to multi-step warming and one-step warming, as one-step warming improved implantation rate according his study, ongoing pregnancy rate on the 10 weeks, also, this is independent than maternal age, pre-implantation genetic testing, and day of blastocyst development and embryo grade. Also, he find out this reduced miscarriage rate and one-step forming, according to him, increase odds of pregnancy by 26%. Of course, on the clinical details, that is a very cool picture, but we have to go on the further slide more metabolic and transcriptomic to see if there is real source on the base that clinical outcome or clinical increasing is right way for us. So as a clinical benefits of ultra-rapid freezing and warming, the ultra-rapid warming protocol, of course, simplify the laboratory technique for the warming oocyte. As a cleavage embryos, as a blastocyst stage, zygote stage, any kind of stage of the embryo, from egg to day 5, you can freeze and warm with ultra-rapid freezing and warming. So this technique reduce unnecessary time for risk of room temperature or stress which is happening during the warming, because every laboratory has a different environmental condition. So some of them have really strict for that, but some of them doesn't establish exactly, especially from the lab air, humidity, and temperature perspective. So that's why to shorten this time, which we are keeping biological material as oocyte, sperm, or egg out of incubator or out of laminar flow of cabinet, so it will be advantage for better outcome. Also, the ultra-rapid warming protocol, of course, the basically risk, there is a risk of the micromanipulation depending of experience and hand skill of embryologist. But we are having this as an egg bank. We are sending on many different countries and clinics, even some of them just starting to have experience for oocyte thawing. So that's why the hand skills of embryologists can be limited, but with ultra rapid warming and vitrification, so this is also we can avoid. And the one of important information that also about ultra rapid warming, we can keep high zona birefringes and zona permeability. So this is also part of our study. So we will measure zona birefringes and permeability, and also the spindle size, location, and morphology once we thaw after the fast ultra rapid warming. And of course, it creates easy workflow, especially for egg banks, especially who has many egg pickup per day, many embryo freezing per day. So that will give an advantage because you can freeze in a 1 minute and you can warm in a 1 minute. So this is actually will be a game changer in IVF labs. Of course, there are also limitations. So we have to focus on also some limitation for ultra rapid. As a basic limitation now, there are limited studies, only 9 studies, like who can give information about, especially on the embryo stage. Of course, that's one of the disadvantage. Also, the poor quality embryo may not survive. This is also hypothesis. Actually, that our experience is showing different. That even for poor quality of oocytes or embryos, that we get high survival. So that is still questionable, need to more deeply study and big number of the sample size. As an objective, what we did in our study, the goal of our study was to check non-invasive. We want to evaluate a metabolism and secretome of the human embryos, which is undergoing ultra-rapid vitrification and morphing. We used mild infrared spectroscopy and also RNA sequencing to identify different biomarkers of the healthy embryos. So as a study, we made firstly sample collection. As our sample collection, like 1,214 donor oocyte, which we used as a sibling way with the ultra rapid and standard vitrification and warming protocol. Of course, after the ultra rapid and standard vitrification, we use intracytoplasmic sperm injection for fertilization, and we made embryo culture with the one-step media till day 5 and day 6 stage. And then we make secretome profiling, which means that lipid profiles, nucleic acid profiles, and protein profiles on the different stage of embryo as a day 1, day 3, and day 5. Also, we made the metabolic profiling on the day 6 during the culture media. And we calculate as IVF outcomes as an oocyte survival rate, fertilization rate, day 3, blastocyst embryo rate, and also euploid rate. And the final stage was RNA analysis for our study. As we always have difficulties to collecting data, especially regarding long-term clinical outcomes, this is the biggest risk of IVF, as we know, because to collect this data and to keep this data from the multicentral function is not that easy. That's why on the cell stage, always metabolic and transcriptomic data will be very valuable. That will show us if we can use this technique or this method on the clinical perspective, and we can accept these new technologies as an implementation to our laboratory. That's why here we use the Fourier transform infrared spectroscopy between the group of the samples as a conventional and ultra-rapid vitrification and warming techniques. And we try to identify if there is some difference between the references of different groups. As you see here, we checked DNA concentration, also the saturated lipids, cholesterol ester as a triglyceride, nucleic acid structure, protein concentration, and the protein structure. What you see on the graphic as a black and the purple graphics, so one of them is showing regular vitrification, another ultra-rapid vitrification. So as a name, if most of you probably on during your master thesis, PhD thesis, you use the spectroscopy. So both graphic, as you see, they're very closer to each other. So that means as nucleic acid especially and the protein concentration, which we are more focused as a lipid content as well, because they are keeping for the stress factors on the development stage of the embryo and oocytes. So they are very closer to each other, and there is no significant difference if you thaw oocyte or embryo with ultra-rapid or regular vitrification and warming you will find out the same metabolic profile. So this is very valuable for us, and this is getting like an advantage for us and avoiding all possible metabolic risks for the development of the embryos from the day 0 till day 6. As our preliminary study for the transcriptomic base, so as we know that transcriptomic analysis give us an advantage, which we can determine the cell based of the stress on the molecular level. So we used here fresh cycle, as we are calling ultravitromics as an ultra rapid vitrification and warming, and conventional vitrification. So you can say that the group size is only 8. Yes, that is right. But you need to understand that for the transcriptomic analysis, it's a really difficult and complicated. So here to analyze and to get the gene expression from especially from RNA base, it's really difficult, complicated, and expensive. So that's why even to achieve that, to analyze as a single gene RNA, that will give us very valuable data. Of course, the next part of the study, I will show the further slides, we will increase the sample size. So here now we worked from the 8 samples from oocyte and blastocyst for each group. As I said, here our aim was to get transcriptomic analysis and to find out cell-based of the stress on the molecular level. So this transcriptomic method give us ability to find out if there is difference between the conventional technique and ultra-rapid technique as molecular stage. The transcriptomic mapping will also give us idea regarding possible side effects for the conventional and ultra-rapid technique. And from this perspective, we can proudly say there is most valuable data we have on the single cell stage for vitrification and warming. As you see here, 98% of the reads pass from the quality standard criteria, which is very high. And here, number of reads range was from the 20 to 50 million. So that means the data is very accurate. So here you see that there are different groups as a fresh cycle, ultra-rapid vitrification, and also regular vitrification from the trophectoderm base, trophectoderm biopsy base, and also oocyte stage. We try to analyze their transcriptomic data. And the principle of this analysis was we performed as a matrix which normalized the gene expression value. And here each column represents a different sample. So here the total transcriptomic data test can be done on the single stage phase. And of course, there is many limitations and very difficult to make this validation. But we succeeded to do that. And actually, our geneticists made a big effort and succeed in building transcriptomic data for this dataset. As you see here, so the different colors, which colors are closer to each other, it shows they are transcriptomically similar. So on the oocyte stage, as you see, 3 different colors are together. Also, the same thing you can see for the blastoma biopsy. So the different color became together. Of course, normally that oocyte as an example and trophectoderm as a sample on the different part of the diagram, but that is very normal because they are different type of cells. As here, our main was actually to find out effectiveness of ultra-rapid vitrification. So the different development stage of the embryos, so different gene can join the process as a normally, and those gene can get different roles. So the graphic show us fresh, conventional, and ultra-rapid techniques show similarity from transcriptomic perspective. And another graphic, as you see, also this is showing us the heat map, as we are calling this is a heat map analyze. So we are using darker color here, means the closer samples. As heat map analyzes, we can see for the oocytes and also trophectoderm based of cells, there is a comparison regarding the gene groups as each two groups. So that means we compare with conventional vitrification and fresh oocyte, ultra rapid vitrification as a fresh oocyte. Also, we combine like conventional oocyte and ultra rapid oocyte. So between each group, we try to understand if there is a similarity as a heat map analysis. So it shows while both group are showing that, as you see, the dark color as a dark blue color, that means that each group and the different colors like orange, blue, green, showing a different sample size. So that means there is no significant difference as a transcriptomically between conventional vitrification and fresh oocyte. That means partially the conventional vitrification and warming was also uh, accurate without any transcriptomic risk and metabolic risk. But the same situation was also for ultra rapid. That means either we are using regular vitrification, either we are using ultra rapid, there is no significant difference for the transcriptomic stage than fresh. So the both technique can be applied in our daily practice without any risk and any problem. So this is very valuable because if both data are giving us similarity for the transcriptomic base, that means there is no further risk for embryo development also for after the live birth. So I am talking about transcriptomic stage, of course. The chromosomal stage is different. Please don't get wrong that. As a time from time perspective and also the laboratory perspective from the efficiency side, it's a big advantage, of course, to use ultra rapid vitrification and warming. And the last graphic, we use also the different techniques as a calculation to calculate distance between each group. So the findings should have, of course, reliability here for the analysis. And for that, we used Pearson pairwise distance analysis for each group, and we try to categorize each group. So when we calculate the distance, as you see, this smaller value, which indicates the closer groups. So when the value decreasing, that means groups are coming closer. So also this analysis show us differences for each group for the transcriptomic data show us similarity. So that means also there is no difference between fresh vitrification and ultra rapid vitrification and warming samples for pairwise distance analysis. So this is also very valuable on the transcriptomic stage. What about IVF outcomes? So if we talk about IVF outcomes, so ultra rapid vitrification and warming comparing standard vitrification, which we evaluate 531 eggs by ultra rapid vitrification, 634 by standard vitrification and warming. So we already warmed them with ultra rapid and standard, so warming, and we find out 100% of the survival rate with ultra rapid warming and 94.5 with standard warming. So this we did totally different quality of egg, different stage of freezing, but of course we follow timing of vitrification post-trigger 38 hours for all group. As a fertilization rate, of course, we find out normally higher fertilization rate on ultra-rapid vitrification because of higher warming rate, survival rate. The same situation on the blastocyst rate as well. And euploid blastocyst rate, there was no significant difference because normally, like as I said, this technique will not improve on anything on the chromosomal stage. Of course, the blastocyst rate and our efficiency will increase, but this is depending on survival rate, not because of this technique is giving more possibility or capability you will receive better quality or more blastocyst. As on the graphic stage, also you can see like ultra rapid vitrification and morphing is showing high efficiency. As a conclusion, the ultra rapid vitrification and morphing, of course, allow the faster cell dehydratation and rehydratation, so which give us higher survival rate and higher clinical outcomes comparing standard protocols. Of course, shortening of the time for the vitrification process, it gives us advantage for the reducing cryoprotectant exposure, because still this is a big question as a cryoprotectant toxicity, how it affects further embryo development, because we are finding out, especially for donor egg, the cleavage arrest rate is very high. Embryos are developing till day 3 perfectly, and then day 3 they stay as day 3. They are not reaching on day 5 or day 6 blastocyst stage, which partially can be connected with the cryoprotectant toxicity or exposure. So the one-step rate rotation protocol, of course, allowing faster rate rotation result. So this is consistent for higher survival rate, and this may directly increase the cumulative blastocyst outcome. I want to add here, of course, that after we are receiving such transcriptomic and metabolic data, we want to increase the sample size and to develop this technique much efficient for everyone and much safer, of course. That's why we applied for European Union as a Eureka project management platform, and we funded 1.2 million by European Union. So our aim now as to increase the sample size for the oocyte, 30 sample for each, and to get fully transcriptomic and metabolic data, including morphokinetics of the zona pellucida, as also morphokinetic of embryo development, which we will record by AI and time-lapse system. Also, we will determine spindle morphology and spindle appearance. Same thing we will do for the embryo stage, as 70 samples for fresh, conventional, and ultra rapid. And the final will be the device validation. So our aim to create a friendly user which is not considering the financial cost or expenses for the clinic, but which will increase the control, especially that device we are planning to have. The AI video recording will be simple, smaller. Every lab can be used easily under the microscope, so they can also control as by the embryologist, not just by the equipment, but equipment will give advantage to create a stability for the temperature, especially to avoid evaporation risk, also to control timing exactly with the different sensors we are planning to do. So that was funded by European Union, and we believe that in final of 2 years, we will come to you also with this equipment and device, which we believe to help for all of you and to keep standards. And of course, all of us have a final aim to help our patient as a cost effective, also with high outcome. So thank you very much for your time and for your attention. I would like to just ask from you, if you have any question, just raise your hand and our moderator will give you a chance to ask your question, and I'm ready to give answer for your questions. There are some questions I see on chat. Till you get unmuted, I can give answer. The first question. So the rapid vitrification, each in equilibration and vitrification solution, at how many oocyte can be or embryo can be vitrified. So basically, when we start to use this technique, I just, of course, increase number of oocyte step by step. If you get the 6 oocyte in same time to put in equilibration solution as a 1 minute, And to aspirate all these 6 oocytes, of course, this is depending again your hand micromanipulation. So always in IVF, especially on the vitrification and warming, the faster manipulation will give you more advantage. So the 6 oocyte, you took it after 1 minute from equilibration and to wash in vitrification for each vitrification 15 times, and then 2 by 2 to load the straw will not give any damage. So here, vitrification solution, one thing that in our mind, which was, as for me, wrong, we always say that to keep in vitrification solution more than 1 minute is very harmful. But this study showed me, so it was not right. So here, equilibration is more critical than vitrification. So that's why we can be more flexible in vitrification solution than equilibration if we are using ultra rapid warming or ultra rapid vitrification. So here one more question about the culture media of high protein. So actually, like many of the clinic, also in our standard study, we were using part of them with high protein, part of them with regular culture media, one-step media. If you ask me if we saw really significant difference, but no, I didn't. But from the high protein media, we receive more blastocysts. Of course, that can be connected also because exactly you cannot choose to select the same morphology or same quality of oocyte between the groups. So I'm not sure how much connected, but we feel that if we keep them on more protein, so during ICSI we feel much better. So morphologically they are looking much closer to fresh.
Hi, Birol. Thank you for your presentation. Just have a couple of questions.
Yeah, sure.
You said that there is more flexibility in vitrification than equilibration. Have you tried to go more than 1 minute in the equilibration solution?
Yes, till 2.5. Till 2.5, and we didn't see any difference on survival. We didn't see any difference on also the embryo development or blastocyst outcome.
Now, regarding the eggs, I have actually started to do some trials here with immature eggs and all that stuff. Obviously, I don't have an egg bank to to play with a good quality material. What about the egg oocytes that were vitrified classically? Obviously, a lot of clinics build up a lot of. I've seen that they seem to be more soft and more granular.
So the firstly, like I also mentioned on during presentation, yes, while we are freezing with the regular vitrification, after warming, even after 1 hour equilibration, we see them more granulated. For ultra rapid, it's a difference. Even that after 5 minutes from the warming, even right after warming, you will not notice that. So if you freeze even with the regular vitrification, you can easily thaw with the regular warming. And one of the advantage also here, doesn't matter which brand of media you freeze those oocytes. It can be Vitrolife, like Cryotech, Irvine. So if you thaw with only TS as an ultra rapid, they are surviving. Because that was also question. I had this problem while we were working in egg donation center, surrogacy center, and we were receiving many embryos from abroad. So especially when once embryos were freezing or oocytes were freezing with Vitrolife, definitely we should thaw with Vitrolife. Somehow it was not working with Kitazato or Cryotech and Irvine. But now I am checking that we made also study. We freeze with a different medias and we thaw all with the same ultra rapid, and also we find out there is no issue for the survival. So that means with ultra rapid, doesn't matter which technique you freeze oocytes, still you can easily apply ultra rapid forming.
What are the timings around the before vitrification and after thawing?
So here is the point. When you do egg pickup, so the point is for vitrification is post-trigger 38 hours to complete cytoplasmic maturation. because those time is very important. Also, spindle take exact location, exact morphology, so that give us more possibility to don't freeze premature or post-mature oocytes. So that's why 38 hours, if you keep as a vitrification, that will be valuable for better outcome.
And after thawing, how much would you leave them in high protein media before ICSI?
Till ICSI, like 1 hour. I generally keep 1 hour. So there are also the question in chat I want to give answer. So the zona thicknessing, that the question is if we observed. Yeah, like as I said, the advantage of ultra rapid is a more closer to fresh oocyte. So also as a real-time picture show us after the ultra rapid forming, we are exactly getting like a similar picture, similar image to fresh oocyte. Because this is one of the issue, like as an egg bank, we are taking a picture before freezing for each egg cohort. So we know that which oocyte we are freezing exactly, and we are keeping that as a database. But when we thaw or when partner thaws, they are sending us oocyte as a warming. So it looks like as a difference, like a morphology a bit changing. Even the color appearance and everything is changing, cytoplasm type of changing. But after the ultra rapid, we definitely faced off that it's totally closer to fresh. So this is also one advantage. As one more question, can you repeat the rapid vitrification? So again, that we have two basic solution as equilibration and vitrification solution. On the equilibration solution, we are keeping as 1 minute, strictly 60 seconds to wait exchange of cryoprotectant and water. And then we are taking them on vitrification solution. I suggest to wash in each solution, each vitrification solution 15 times the oocyte to avoid this equilibration part. And definitely you need to change pipettes between and then load the straw. So the question after using ultra rapid forming, how much time will it take to expand? As I said, for my experience, the expanding time is so quick. Even right after warming, if you go to check under microscope, you will see that they are totally expanding. But of course, we have to wait that still partially cryoprotectant will be on perivitellin space. So that's why we are keeping in a 1 minute, 1 hour on the culture media in incubator to also wait a total expanding. There is question if we have experience with M1 oocytes. As I said, of course, this is a study, and for as a preliminary study, we work with the M1 oocyte, the GV oocyte, very low quality as a morphological of oocyte, any kind of the morphologic part we worked, and that's why that we are easily saying that ultra rapid vitrification and warming, it is very secure. Especially you are asking for patient with who has a cancer treatment. Of course, I understand very risky for them. They have several oocytes and this is their future. And I believe that ultra rapid vitrification is a more effective and secure for them, especially on if you try on day one zygote, if you are freezing, or day 3 stage, and also you will see that embryos are looking really good after ultra rapid vitrification and warming. So what is the the risk of TS exposure if it stay longer and it doesn't detach from the carrier? This is also like I can say that our study shows, of course, we are trying to keep 1 minute strictly because this is not just for survival. I mean, if you pass the 1 minute, still oocyte will survive. But as you see that transcriptomic and metabolic data, once we pass 1 minute, the data is changing a bit. So this is a bit partially risky. That's why I prefer always to keep the 1 minute. Okay, I guess no more question. If you don't have any more question, I would like to say many thanks to join Anyway, we are all colleagues. We know each other. Anytime you have any questions, so I'm always open. We can share information between us. If you have any experience, I wish all your experience will be good, but an experience, if you share with me, also, I will be happy. Anyway, on the clinical based of study, the second part, we will go to multicentral study. And of course, any of you want to be part of it, it will be our pleasure to accept you to be part of our project.